I'm excited to kick off this event today and tell you a little bit about our work. Many cells in your body reside in an intricate network of proteins called the extracellular matrix, or ECM. You can think of this sort of like the cell's home, providing protection from the elements, as well as a place to store all of the important things that help them to function. Certain cells reside inside tissues like tendons and ligaments, are subjected to high mechanical forces every time we move our joints. In order to withstand these forces, cells have become very good at keeping their home in tip-top shape through a process called ECM remodeling. Cells sense changes to their environment through the extracellular matrix and then repair and remodel tissue structure in order to improve function. We study this process in my lab using whole tendon explants, which enable us to have the entire cell structure along with their home and subject them to things like exercise or different biochemical factors. We can then watch as these cells degrade and repair the matrix in real time so that we can understand what factors are important to this process and how to make it more efficient. Importantly, we have discovered that this process of ECM remodeling is actually dependent on sex. When we subject tendon explants to a loss in activity, such as disuse, we see that the males are much more active than the females. They have increased matrix synthesis, like collagen production, as well as degradation, suggesting overall a more rapid or robust turnover of ECM proteins. While this paints a picture of female tendons being a little bit less dynamic than male tendons, we actually know that that's not really the case. Females disproportionately suffer from a number of degenerative disorders associated with chronic ECM breakdown, including, as you can see here, carpal tunnel syndrome and osteoporosis, which are approximately 4 times more likely in women and preferentially affect older women. So this led us to question what other variables we might be missing that could influence ECM remodeling. And perhaps unsurprising to this crowd, we started studying sex hormones and their response in ECM turnover. It turns out that when you add in single hormones like estrogen or progesterone, you're able to rescue this decrease in protein synthesis and even further increase it above that of males without affecting ECM degradation. So what does this all mean? It means when we have natural drops in hormones, such as during the menstrual cycle or in menopause or aging, our tendons are not as able to adapt to different changes in mechanical stimulus, and thus they leave them susceptible to injury or degeneration. Importantly, we found that these hormone effects are dependent on whether you're looking at male or female tissues, whether these cells are exercised or not, or whether you're looking at single hormones or multiple hormones delivered together. Since sex hormones rarely ever occur in isolation inside the body, we think these hormones are really important to understanding how they influence ECM turnover. And so we've been working hard towards mimicking natural hormonal fluctuations and interactions in the lab by recreating the menstrual cycle in vitro. We've been building a model system to integrate macrofluidic fluid control so that we can recreate the temporal dynamic and range and also the plethora of hormone interactions that we see during the natural menstrual cycle. We hope to build an accessible model of female-specific biology in order to study mechanobiology research and aim to use this model to test different therapeutics or drugs so that we can figure out how to keep women healthy and active well into their later years. So if you have any questions, talk to me later, and I hope you enjoy the rest of the talks.